In this video, we're going to be discussing how you can find the Km, the Vmax, and the kcat from kinetics experiments. And so by the end of this video, I want you to be able to do the following. I want you to be able to explain how an enzyme kinetic experiment is performed. I want you to be able to look at data from an enzyme kinetic experiment, and from that data, be able to derive the Km, the Vmax, and the kcat. I want you to be able to describe what the Km, the Vmax, and the kcat tells us about an enzyme. I want you to be able to define what the catalytic efficiency is and explain why it's an important characteristic to know about an enzyme. And finally, I want you to be able to explain the concept of catalytic perfection. Okay, let's start off discussing how one would perform an enzyme kinetics experiment. And the idea here is that it is an incredibly simple experiment to perform, and there's a remarkable amount of information that you can get from it. If you were to take a biochemistry lab and wherever you end up transferring, I guarantee that one of the main things you will be doing is an experiment like this, an enzyme kinetics experiment. And this is how they're performed. What you're going to do is set up a whole bunch of tubes, a whole bunch of reactions. Okay, so let me first say E is the enzyme that you are testing. S is the enzyme's substrate. Okay, so you are basically looking at adding that enzyme, adding substrate, letting that enzyme bind substrate, turn it into the product, and then release the product. Okay, so you will set up a bunch of tubes. I am only showing 3 here, but these ... mean you're going to be setting up more than just 3 tubes. The important thing, in each tube, the total amount of enzyme that you add is the same. Absolutely essential that you understand that concept. And then to each tube, you are going to be adding increasing concentrations of substrates. So substrate concentration 1 here, substrate concentration 2 here, substrate concentration 3 here. So really the only thing that you're changing from tube to tube to tube to tube is the concentration of the substrate that you are adding. Now with each tube, you are going to be calculating. So the data that you're going to get is the initial velocity, the micromolar of product that is produced per minute. That's your initial velocity. So you're going to determine it for each one of these tubes that you set up. So then you have 2 pieces of data that you're going to plot on a graph. You have the substrate concentration and the corresponding initial velocity. The substrate concentration and the corresponding initial velocity. And you will make a graph that looks like this. So here on the y-axis is the initial velocity. Here on the x-axis is the substrate concentration. And what you will see is you'll get this hyperbola right here. And you'll get to a point where it just about flattens out. Okay? And from this data, you can determine the Vmax, the Km, and the kcat. Let's figure out how to do that. The Vmax, remember, the Vmax is going to be the highest initial velocity that you see. Basically, that's going to be the asymptote to this hyperbola. So right here, this initial velocity, the highest initial velocity where our hyperbola starts to sort of flatten out, that is the Vmax. Now, to find the Km, remember that there's this special relationship between substrate concentration and Vmax and Km. And at 1/2 Vmax, so let's take a look at the initial velocity that's equal to 1/2 Vmax. So here's Vmax, and here's 1/2 of that. So there is the initial velocity that is 1/2 Vmax. So then you figure that out, you go, you hit the curve, and then you figure out, well, what's the concentration at that initial velocity? So that is the Km. Okay, so the Km is equal to the substrate concentration when the initial velocity is 1/2 the Vmax. Got it? All right, finally, kcat is something you have to derive after figuring out the Vmax. Okay, and the way that works is you figure out the Vmax, and the Vmax equals the kcat times the total amount of enzyme that you added. Okay, so this is sort of a little independent of this graph except for finding the Vmax. So from this data, we can find the Vmax of an enzyme, the Km of an enzyme, and the kcat of an enzyme. Now, a problem that we have when we look at the data this way is that this hyperbola never actually reaches the asymptote. So you don't actually know really easily what the Vmax is. You sort of are estimating it. To solve that problem, Lineweaver and Burk sort of modified the Michaelis-Menten equation such that when you plot it out, it makes a line. And what they did, they say, well, if we look at 1 over initial velocity equals Km over Vmax times 1 over the substrate concentration plus 1 over Vmax. If you write the equation this way, then what it looks like is y equals mx plus b, the equation of a line. So that now, instead of mapping out or instead of graphing out the initial velocity as a function of substrate concentration, instead you do what's called a double reciprocal plot. You plot 1 over initial velocity and 1 over substrate concentration. And when you do this, you are going to get a straight line. Okay, now let's figure out how to find the Vmax, the Km, and the kcat from a double reciprocal plot. Well, the Vmax is right there. Well, that's actually 1 over the Vmax. So to figure out the Vmax, it's Vmax is 1 over the y-intercept of that line. The Km is right here. So to find the Km, well, this is 1 over the Km right here. So to find it, it's the Km is -1 over the x-intercept. And then finally, to find the kcat, you do the same thing we did in that previous slide, which is the Vmax is equal to the kcat that we, Vmax, which we found, is equal to the kcat times the total amount of enzyme that was added to the reactions. This is how you are going to see enzyme kinetics data most of the time in many of the problems that you're going to see in your book and on the exam. So it's worth understanding this, but make sure you also understand it this way as well. All right, now let's take a look at what Km, kcat, and Vmax tells us about an enzyme. Km is a measure of the enzyme's affinity for its substrate. Okay, how well does that enzyme bind to its substrate? Now, it doesn't measure how fast that enzyme increases the rate of a reaction. It simply tells us with how strong of affinity does it bind to the substrate. And the lower the Km, the stronger its affinity for its substrate. Okay, that's a super important thing you need to know. The lower that Km, the stronger the enzyme is, has an affinity for its substrate. Now, kcat is sort of the opposite. Now, the kcat of an enzyme doesn't tell us how well it binds to the substrate, but what it does tell us is once the enzyme is bound to the substrate, how fast is it going to catalyze that reaction? Okay, and the higher the kcat, the faster the enzyme catalyzes that reaction. Now, the important thing to remember, the Km and the kcat, they are inherent characteristics of the enzyme, independent of enzyme concentration, okay? Which brings us to Vmax. Now, Vmax is equal to, as we saw, kcat times the total concentration of enzyme. Basically, when all of the enzyme that's in solution is actually bound to the enzyme, That gives us the Vmax. And the Vmax is the fastest an enzyme can act at a particular enzyme concentration, which means Vmax isn't something inherent of the enzyme. Vmax is dependent upon enzyme concentration. If you increase the concentration of the enzyme, you're going to increase its Vmax. Now, there is a calculation we can do that gives us an important constant, and that is called the specificity constant, or sometimes called the catalytic efficiency. And this number gives us a way to compare one enzyme to another, because here's the problem. There are some enzymes that have a very, very high kcat, meaning they're really fast, but they have a really, really high Km as well, meaning they don't bind very well to the substrate. Okay? Or there's some enzymes that bind really, really, really well to the substrate, but once bound to the substrate, they don't speed things up very much. Okay? So these are enzymes that are kind of really good at opposite things. What's nice is when you take the kcat and you divide the km. So basically when you kind of include both of these features, kcat and km, into the definition of that enzyme, or characterizing that enzyme, then you can start to more accurately compare one enzyme to another. Because when you compare the kcat divided by km here between catalase and fumarase, they both have similar efficiencies. Now, they get to those efficiencies in two very different ways. One has a very high kcat, one has a very low km, but their efficiencies are fairly similar. Now, I got this question for you to think about. The blank the specificity constant, so higher or lower, the higher the efficiency of the enzyme. Take a moment and think about it. Okay? Hopefully you've paused and thought about it. Well, let's take a look. The faster an enzyme is, the higher its kcat should be. Good, and that's in our numerator. The more affinity an enzyme has for the substrate, the lower the Km is going to be. That's on the denominator. So when you divide a big number by a small number, you're going to end up with a big number. So the answer here is the bigger or the higher the specificity constant, the higher the efficiency of that enzyme. Now, there is a limit to just how efficient an enzyme can be, and that idea is called catalytic perfection. In order for an enzyme to catalyze a reaction, it must bind to the substrate. And enzymes and substrates are dissolved in an aqueous solution, and things bouncing around within an aqueous solution is limited by the diffusion rate of the enzyme and the substrate in that aqueous solution. And you can't go any faster than that diffusion rate. So that diffusion limit is something about between 10 to the 8th and 10 to the 9th over molar seconds. This represents the maximum efficiency of an enzyme. All right? And that's called catalytic perfection. So if an enzyme approaches the diffusion limit of an aqueous solution, then we say it is a catalytically perfect enzyme. And if we take a look, some of these, here's our efficiency, catalytic efficiency, kcat over km. Look at that, 1.6 times 10 to the 8th, 2.8 times 10 to the 8th. That's all approaching this diffusion limit here. And so these are approaching catalytic perfection. All right, there's a lot of very practical stuff in this video. Watch it over and over and over again, and make sure by the end that you can explain how enzyme kinetics experiments are performed. Even though we don't have a lab, I want you to know it as if you did do it in a lab. Derive the Km, the Vmax, and the kcat from enzyme kinetics experiments, both a normal hyperbola and then the double reciprocal plot. Describe what Km, Vmax, and kcat tells us about an enzyme. Define catalytic efficiency and why it's an important characteristic to know of an enzyme. and explain the concept of catalytic perfection.